Okay. So onion root tip having a stem cells which grown fresh. Okay. So firstly, this is onion root tip which is having a maturation zone, then zone of elongation and meristematic zone which is the tip of the onion. So method of growing onion root tip is now whenever the fresh onion we add into a beaker with full of water, we add the roots or dry roots of onion. Now the duration is 7 to 8 days to grow the freshly grown onion roots. Okay. So now onion is like that. The thread-like structure is grown into the water. This is your freshly grown onion roots after 7 to 8 days, that is 1 week of duration. Okay. Now what is mitosis? There is having a cell division which have the 5 stages which known as first is prophase, second is metaphase, third is anaphase, fourth is telophase, and last one, that is cytokinesis. So we are going to study, there are 5 stages from prophase to cytokinesis. There is 5 stages of mitosis. So first one is prophase, metaphase, anaphase, telophase and cytokinesis. Okay. So we are going towards the first stage of mitosis, that is prophase. Whenever the cell is start dividing, the prophase is initial phase of mitosis. So in the initial phase, the nucleolus chromosome is distinct in the nuclear membrane. Okay. So there is a complex structure in prophase phase. Okay. So in metaphase, there is a sister chromatid is in equatorial plate-like structure. Okay. So as we see here, the spindle fiber get attached towards the sister chromatids. Okay. So they are not separated in metaphase. So in metaphase, the sister chromatid is in the equatorial plate-like structure which is start dividing into the anaphase. Okay. So in anaphase, the spindle fibers get separated the chromosomes. Okay, which is known as the daughter chromosome. Spindle fiber is act as a stretching the separate chromosome, that is a sister chromatid separate towards the each other. Okay. So whenever they separate each other, the spindle fiber help them to stretch out. Okay. So this is your anaphase. Next stage, that is telophase. Okay. So this is the main stage, that is the daughter cell divide. That is start dividing only but not fully divide, which is divided into cytokinesis. Okay. So in telophase, the daughter cells start dividing and having a two separate daughter cells. So this is your telophase. Last stage of mitosis is cytokinesis. Okay. So separation of cytoplasm. What is mean by cytoplasm? That is nucleus. Okay. So separation of nucleus in both daughter cell called as a cytokinesis stage. So this is your overall stages of mitosis. This is your requirement for mitosis. Okay. First requirement is onion root tip. Okay, which we grown by 7 to 8 days grown onion root tips. You have to take 2 cm of onion root tip with the help of forceps. So this is your onion root tip which is freshly grown. Next requirement is distilled water, that you have to wash the onion root tip. Next is ethanol, you have to wash with the ethanol also to avoid contamination. Next is dilute HCl, you have to add dilute HCl and heat it. Next thing, that is acetocarmine stain, which is used for the give color towards the onion root tip. Next is slide, then cover slip, spirit lamp and dropper. So here we take onion root tip. You have to cut 1 to 2 cm like that and discard the further one. Next thing, Next thing that you have to add, firstly water, that you have to wash root tip like that. Wash with the distilled water. After that, wash with the ethanol. Discard water in discarding beaker. After that, add ethanol. Use another dropper for another solution. Add ethanol and wash root tip, that avoid contamination, and after that 1 to 2 seconds you have to discard ethanol also. Okay. Next thing that you have to add a dilute HCl. After adding dilute HCl, you have to hit like smear hitting, just 1 to 2 times. That's it. You have to give a warm hitting to root. Okay. Next, discard dilute HCl again. Next procedure, you have to add a acetocarmine solution that gives a stain and color to onion root tip. Okay. Okay. So next procedure, now we have to add some drop of acetocarmine. Now you have to incubate this for 30 minutes to observe stages on microscope. This is microscope. You have to observe mitosis stages under the microscope. Okay. So this is eyepiece. Next thing, that is, this is adjustment knob. This is your lenses, that is 10x and 40x lenses. This is your platform where you put the slide. Next thing, that is inclination joint and diaphragm. This is your mirror where you have to adjust the light. Now the staining is complete towards the onion root tip. Okay. Now you have to discard excessive stain and add this root tip on slide. Take a clean slide and add a root tip. Then put the cover slip on it. So this is your slide. Okay. Now squash the root tip with the help of forceps. So this is your temporary slide of onion root tip. Okay. Now you have to observe under the microscope. Place it on the platform like that and cover with the clip of both sides. Okay. Now adjust the mirror towards the onion root tip. Okay. Now with the help of adjustment knob, you have to zoom and observe the stages which is present in the onion root tip.